This project is looking at Phytophthora sojae. That's the pathogen that causes Phytophthora root and stem rot in soybeans. And they're trying to figure out what's going on with the populations throughout the state. So other people, Kat, Jane, and Dean Malvick are also involved in this project. So just as a reminder, Phytophthora root and stem rot lesions typically begin below the soil line and then move up the stem. This is considered a chocolatey brown color, but the pathogen can infect the plant anytime during its development. You can also see that it causes pre and post emergent stamping off, so seedling disease and dying. And it's hard to distinguish the plants that have died due to Phytophthora from plants that have died due to, I forgot to start my timer, due to species like Pythium. So Phytophthora sojae is considered a water mold, and so it is really favored by saturated soil conditions. So soils with a heavy clay content, soils with poor drainage. Sound familiar? Those soils are at particular risk for Phytophthora root and stem rot. So in the pathogen itself, the pathogen produces what are called AVR molecules. And those molecules it releases when it infects the plant. And these molecules try and disarm the defenses of the soybean plant. But one of the main ways that we actually manage Phytophthora root and stem rot is through variety selection. So what we do have is a series of resistance genes called RPS genes. This is named for resistance to Phytophthora sojae, so RPS. So if the plant has a complementary RPS gene to the pathogen's AVR gene, it's able to know that the pathogen's there and mount a resistance response. And so knowing which AVR genes are around in the Phytophthora population throughout the state is really important for knowing which genes would be most effective in our soybean varieties that we plant. So let's see. I had to cut this way down last night. So the entire project here is trying to figure out which of these AVR genes are most prevalent throughout the state. Do we have varieties adapted to Minnesota in maturity groups that we can grow here that are still effective? That's really important information and really important to soybean breeders like Dr. Lorenz here. So this past year, and then going into 2024, they've been collecting both soil and plant samples, isolating the pathogen from those samples, and then what they've been doing is using a molecular tool. So in the past, when researchers used to study AVR genes, what they had to do was plant 10 or more plants, soybean plants of a particular variety with a known RPS gene in it. They had to do that with all of the plants, with all of the known RPS genes that were out there. So 10 plants per RPS gene. That takes a lot of time to grow those plants, maintain those plants. Each of those plants needed to be individually inoculated with the pathogen and then grown up in the greenhouse for a period of time and evaluated. Those evaluations could sometimes give you false positives or negatives. And so there's now a molecular tool that they can use that really is quite accurate. So it has about 98% accuracy. They're not getting that high percentage of false positives and false negatives. And so this tool, it costs a little bit of money to use this tool, but it also tends to have less of an expense in manpower and time. And so that's the real advantage to this particular molecular test. So here's just some results from these molecular tests. So first things first, they wanted to make sure that it's called a multiplex PCR. They wanted to make sure that this worked. So what they did is they took multiple isolates of Phytophthora. They also had the pathogen that causes white mold here and water as a control. And they use this multiplex PCR. So here's the different AVR genes that they were looking for. And all of these Phytophthora isolates, they knew which AVR genes were in these isolates, and this was just a sort of proof of concept. Does this particular assay work well to identify AVR genes that they already knew were in these pathogens? And it did. Each of these black dots here are bars. That's showing you that indeed that there is that AVR gene there. So this is working well. They have it up and running, and it's going to save tons of time and money. So these individual counties in red and purple were the counties where samples were submitted this past year. So we had a farmer down in Norman County that was out scouting the field one day and noticed, wow, there's all these plants dying. Had those typical chocolate brown lesions, and we sent in samples, and what was neat is they're currently isolating the pathogen. They're currently using this molecular test to figure out which AVR genes are there, and they're going to share that information with the producer. And so this is a really unique opportunity if you have Phytophthora root and stem rot show up in 2024 to get your own Phytophthora isolates tested to see which varieties would work best for combating Phytophthora sojae in your field. So there are a lot of people involved in this trial. Here's Dr. McKay's lab, Dean Melvick's lab, Dr. Sanyu Chen, his lab, and then this is part of a larger multi-state study. So North Dakota, Wisconsin, and Ohio are all doing a similar survey throughout the state. Please contact Megan McKay, Kat. I think I have some information about how to contact them. If not, it's in this research book. If you have a suspected Phytophthora population or Phytophthora infection in your field.